can I just say as well, we are videoing, so if you can, I don't think the mic's brilliant on that, so if you can speak up, then that would be brilliant. Thank you.
And remember to smile.
Work package 1 is about, how does that work?
Oh, it's the other one, sorry. That one.
Right. It's divided into different tasks. I'll try to go through them, or most of them, at least. The first one is about spectroscopy. That means a different way of analyzing seed, mainly about seed-borne diseases, but also about seed vitality. It's led by Johannes Ravn Jørgensen. He's not here today, as far as I know. And also, I'm not sure about really Søren Rasmussen, whether he is actually in work package 1 or work package 3. Because he's not working with seed-borne diseases. He's working with sorting according to protein content. And he hasn't really started his task yet. So we can discuss that later, whether he actually belongs in work package 1 and work package 3, but he hasn't done anything yet. And it's very poorly described in the project description what he's actually going to do. But he has a sorting equipment for sorting seed according to protein content, and I'll go back. I've used this equipment, so I'll come back to that later.
Anders, can you speak up, please?
Yes, I will try. Johannes Jørgensen is working with seed analysis. He's trying to use novel equipment to see if we can actually detect seed-borne diseases on seed. He used different equipment. It should be maybe possible to...
No, you have a pointer on this thing.
Yes. Does the internet work here?
I haven't tried it.
Should work for that. Yeah.
It should do.
Maybe it does work. Let's see. It's just to say that normally when we analyze seed-borne disease, we'll put it on a piece of paper and some water, let the fungi grow, and then we can analyze the fungi. What this equipment does is that they illuminate the seed, the dry seed, with different spectras, and then it's quite advanced equipment, so they can actually see things that we cannot see with the naked eye. So they found out that at least Fusarium works quite well, but also other diseases, that they can actually see these infected seed among a sample of healthy seed, and that could make seed analysis quicker and easier, and thereby cheaper, which would be quite good for seed analysis for organic farming. He also used NIRS, that means near-infrared. This equipment has a lot of different specters, hundreds of different specters, including the near-infrared. But this is a reflection, but he also works with near-infrared transmission. He is also not very far in the COBRA project. He had a student from Slovenia, I think, a master student making the analysis in Denmark with Slovenian seed. But he's also calling for you to send him infected seed, so he actually has some material to work with to make these calibrations for this equipment.
Does he need seed that is defined, the infection, or not?
Yes, defined, yes. The next task, please feel free to ask. I'm not promising I can answer, but the next task is my own. I'll not go much into detail with this because I also have a poster showing the same thing. What it is about is that that common bonds, like many other diseases, is not a defined thing. It's divided into different virulence races. So if we want to find out if a sample is infected, it has a resistance gene, then we need to which variety has which resistance genes. For example, in this experiment, I have tested a number of varieties for common bond infection, and some are susceptible, and some are. Is this the pointer? You can see that the top up there is the high infection and some resistant down here. But that is when I take healthy seed, put on some spores, and see the infection level. But that's not the situation for normal farmers. Farmers, they will take infected seed and sow them in their field and get infection. So the spores will come from the same variety. Whereas in this experiment, the spores come from some other varieties. So if I test the same varieties with their own spores, as is the practice in normal farming, the picture looks completely different. Suddenly, many of the infected varieties, many of the resistant varieties actually becomes susceptible because we have virulence against this infection, against this resistance gene. So a resistant variety is not really resistant when it comes into farmer's field. And then if I take some of the apparently resistant varieties but with actually virulence and take this one and infect the other one here, then cross-infecting the different varieties, then I can actually see if I get virulence in another variety, then apparently they have the same resistance gene. I hope. Then it looks like this, that I can see here a group of varieties infected with spores from different varieties, and they react in a similar way. So I take that as a sign that they probably carry the same resistance gene. And next year, I've included some varieties with known resistance gene, and then I get the idea that this group is actually reacting in a similar way to different spores, which means they probably have the same resistance gene, and since one of them have the resistance gene BT2, then I think this group of varieties probably has the resistance gene BT2. That's a hypothesis, so this is what I'm working on. And you can see my poster tomorrow, or later today. Next task is the task in Austria. They're actually working with the same idea, but they take another approach. They use genetic markers to try to identify resistance gene and virulence races. They have a PhD student working on that, which is very good. So what they do is that they cross different varieties, susceptible and resistant varieties, a group of resistant varieties crossed with a couple of susceptible varieties, and they are not working with common bond, they are working with dwarf bond, which is they have the same resistant mechanisms, but it's two different species, and they react. Dwarf bond is mainly a soil-borne disease, whereas common bond is a seed-borne disease. Both are both, but mainly. And they. Right?
Yeah, annually.
Their main problem is that dwarf bond is a serious disease some years in some places, but not elsewhere. So they have quite problems with having good infection sites for their experiment. So they're trying it in different environments, and they also send some to America because they have very good infection over there. They also send it to Germany and Sweden. But it's this. I think these draft one researchers are cooperating quite well, and in different locations also in Austria, and especially one of them had very poor infection. So they will also try to do something in common bond, because it's the same resistance system, but common bond is much easier to work with. In Germany, Berta Killermann, she's not here, is she? I don't think she signed up for this conference, but she has a PhD student working also with dwarf pond. They mainly work with the infection in the soil, so they are trying to extract spores from the soil to find out whether this field is in risk of dwarf pond infection. This is an ongoing work that has been done for some years, and they will continue in the COBRA project. And then I think the main task in COBRA is actually as an experimental site for the Austrian research. But at least they work quite with this detection system, and I can see that at least they have someone going in the field this year as well. So they are doing something. I haven't been there. Michael Lunke is not here, is he? I'll not talk very much about this climate change thing, because it's in Maria's work package, most of it, and I think you will present most of the work. And Michael Lunke has a poster here on the specific task that is related to seed-borne diseases. So I will not talk about his work, but our Baltic colleagues in Latvia and Estonia are doing a wonderful work, very active. And, that's your field. Look nice. A couple of COBRA partners have sent seed to Latvia, and you're testing them for seed-borne diseases, susceptibility of seed-borne diseases, and there are also yield trials in barley and I think also winter wheat. We have a good, and the Baltic partners are associate partners, so they're not receiving funds from COBRA. I think we have good cooperation. And they have very good sites for testing winter hardiness.
Yes.
We may send a couple of lines up there to test them for that. To be honest, I'm not very well into the legume, what the legume people are doing. You are.
You want me to summarize?
Yes, please. That would be helpful. Yes.
This is the.
Excuse me. I think it would be great if everybody says their name.
Oh, so
Yeah,
I'm written there, but yes, I'll introduce myself. Yes, I'm Luciano Pesetti from CRA in Italy. What we have done, what we had to do, and what we have done was to carry out two trials in Italy, one in northern Italy, Lodi, the other in central Italy, in Rome, under field conditions, treating the pea seeds which were inoculated with the anthracnose species, the three species, with two different essential oils. So based on preliminary assessments made by my colleague, the pathologist in Rome, he chose the two oils. One is thyme oil, Thymus vulgare, and the other is clove oil, Eugenia calyphyllaea. So the seeds were treated before sowing, after inoculation. The trials we carried out, we just finished. I don't have any results in a tabular or graphical form because we just finished harvesting the trial in Lodi, in northern Italy, because the other trial in Rome was basically abandoned because since the beginning, the inception, for physiological reasons that we do not know, the controls did not emerge properly, so we couldn't use it. And the trial in Lodi itself was not resolutive because we chose two varieties. We wanted to have two contrasting varieties. So we had one commercial good variety which is moderately tolerant, and we asked the same seed company to provide us with a susceptible variety because we want to have a contrast. But apparently the susceptible one, which was an old variety which was abandoned commercially, was extremely susceptible. So whenever there was infection in that variety, the infection itself halted the emergence of that variety. So the effect of the oils on that variety was really poor. What we have decided then with the colleague in Rome was to repeat again the trial coming autumn, because it was an autumn sowing, possibly adding a third variety of an intermediate susceptibility in order to see if we get something which is more representative than this very susceptible, hopefully susceptible variety. What we got as preliminary result from the trial in Lodi was that, well, two things. The treatment with the immersion of seeds in the oil solution was apparently to some extent toxic to the seeds. So that decreased the emergence of the seedlings. Whereas the other treatment, because I should have mentioned, we used two different ways of treating. One was by immersing into the solution, the other was by spraying the essential oil and coating it with a kind of film. That second did seem to work much better, and in particular for the moderately tolerant variety, we got good result in terms of final number of emergent plants. We checked throughout autumn until spring the number of plants that emerged per floor. And we got good result in that one. And also in terms of final grain yield with the thyme oil, which was sprayed and coated. So this is a preliminary result that we would like to confirm, and of course we would like to repeat both trials, both sides, and possibly adding a third intermediate variety. Besides, that was the field activity. Luca Riccioni, the pathologist in Rome, is in contact now with Kevin David, here, from the University of Ghent. So in that case, I'm just a liaison between Kevin and Luca, but they've started thinking of a controlled environment experiment using some of the oils to control Colletotricum. Am I correct?
On looping seeds. So anthracnose on peas is not a problem in Belgium, but if we sow lupins, we have problems with anthracnose and lupins. So we take the same or similar disease, but on another region. That's the reason I chose for lupin.
And finally, some of the seeds that we just harvest will be provided after checking for the disease, the level of infection will be provided to Dr. Jorgensen in task 1.2.1.1 for that screening, visual screening of seed-borne diseases. So we have agreed already in bone whenever we get some of the seeds with the infection level and disease checked, I will provide him with some samples.
Right, thank you very much.
I have a question. Could you please tell us which essential oils?
Thyme, Thymus vulgare, and clove, Eugenia caryophyllea. These two are the essential oils that we chose again after preliminary tests that were made by my colleague in pathology center.
the last task from Jens Knudsen is a very small task. Is screening for resistance to anthracnose. But I don't think that there can be very much cooperation because he's working with spring peas in northern Denmark and you use winter peas in southern Europe. So I don't think that his varieties can be used. His way of doing it is to grow a composite cross population and then pick pure lines from this composite cross. But he's not using the composite cross as many of the others are using it, growing it as a composite cross. He's mainly using it as a gene bank and for natural selection, and then he'll pick pure lines from this system. And I think that's about it. Well, I actually forgot to talk about my own task, work package, task 1-3, but that's because I have a presentation on that in the afternoon, so that'll be a repetition where I should go into detail in that.
And can I just ask, anthracnosis in peas, that's also collated trichum?
No, it's Ascopita and Mycosporella pinodes. Ascopita peas and Mycosporella
So you're
pinodes.
talking Ascopita, like?
Yeah,
Yes. We also in Denmark we call it something completely different.
Okay.
Anthracnosis, okay.
So please use the scientific names because you are causing huge confusion. Because if you talk using a treatment against Ascopita blight and then trying against Colletotrichum in lupin, I assume anthracnose is usually Colletotrichum. And so please use the scientific name.
Yes.
The problem in P is that the pathologists are reclassifying all the two of the three species have been recently reclassified. Now they call it Peyronella pinodes and Peyronella pisi.
Didymella. Didymella pinodes.
Oh, that is a new one.
That's why I'm asking, even if you use an old scientific name,
It's
still know.
Ascopita pisi and Ascopita pinodes.
Right, okay. Let's just see where we were in terms of schedule. Right, we're about 15 minutes behind anyway, so we can have a few quick questions if there's any other sort of questions arising. Otherwise, we can move on to Maria and catch some time up. Any questions? No? Thanks very much, Anders. That's great. Cheers. And at this point, what I'd like to say, which I already kind of mentioned, which I meant to mention at the start, is that everybody has delivered fantastically well on this project so far. So actually, I mean, we're seeing updates. We're only 18 months in, so sometimes it's a bit difficult. It's more sort of methodological, the actual results side, but we're getting more back than we expected, and that was written in the proposals. And I know that Core Organic are very happy with us. So I'd just like to say thank you to everybody for working so hard as well.
Yes, I can say that too, because what I did is I sent out a letter about 6 weeks ago to the Work Package 2 members and just sent them this actually not intended but very useful one-year report that I think you and Henry put together in January, and asked people basically to update on Work Package 2, which basically will serve me as the 18-month report in September. So most of this is already written, because most people have already sent their updates, and I just collated it into this one-year report. So I would like to thank you also for doing this report, because it has been extremely useful to collect information now. Work Package 2 has 3 parts. Study the potential of high diversity, which is mostly composite cross populations and specific genotypes to adapt to climatic fluctuations. Second, to find determinants of early vigor and competitiveness against weeds. And third, to identify breeding strategies for coping with multiple stressors through studying the effects of variable degrees of functional diversity in crops. You may want to turn the mic a little bit if you want my voice. This is for the
Yeah,
guys in
I'm only
India.
pointing at the screen.
There's 3 subtasks in this task. One, it's evaluation the cycling effects in the winter wheat. I'll get to that. Then barley for adaptation to extreme climates. That's Rege van der Jurgensen. I'll show some stuff on that. And evaluation of grain legumes for adaptation. That's Peer Hazard. Then in the second one, early vigor competitiveness. That was Niels Ove, who also sent me some information. So he has looked at the composite process for allelopathy and looking in the root morphology. And the third one is breeding strategies for coping with multiple stressors through studying the effects of variable degrees of functional diversity. We have can we use diversity for coping for stressors, winter wheat composite crosses, and coping with weeds and intercropping. And basically, most of this task 2-3 is incorporated in the other tasks implicitly. So the cycling effects, we have currently 12 partners involved. Actually, I think it's wrong, we have 13 because I forgot to change that number because the Maribor people actually told me they have also some cycling populations in some of the conventional organic and looking at stuff. At all sites, the cycling composite crosses are grown as planned. Then ORC and we have at least experiments with the cycling populations ongoing, and then we have more experiments with the composite cross populations in France, Denmark, Italy, Slovenia, also in the Netherlands and Hungary, although they're only associated partners. This is, thanks Peter. He made a very nice thing about what the cycling really means. In 2005 we had the composite cross populations split. They were produced in the UK and we had these 3 different populations, and one bag for each population was split into 4, and the French, the Hungarians and us received, where are we? 6. We are number 6, yeah. And we received a sample of this, so it all came out of one bag. We maintained the seeds. It didn't really work in France very well for various reasons, but so the Hungarians, the British and us kept this population since 2005, and in 2008 we decided to send one of these populations to other partners and expose this population to variable conditions from year to year. So we are sending our seeds after harvest to the Swiss are sending it to the French to the British to Denmark, to Anders is sending it to Peter in Munich, then it goes to Martin Varsa in Hungary, then it goes to the Netherlands, and then it comes back to us. Since 2008 to 2014 now we have at least one full cycle. No, they're not quite through all the sites yet. But we have different populations that started in different places, so we have a big number of different populations. And you can see this in the poster in more detail. This year, for example, we had a severe stripe rust epidemic in Germany. I also saw it in the Netherlands. I do think in Denmark they also had it. Other places, I know Belgium had it. If the reference varieties were susceptible, the populations were comparatively healthier. Of course, we also have several reference varieties growing. Some of them were nicely resistant, but we also have selected our reference varieties to represent a spectrum of susceptibility, so we can also pick up if a disease is important. And there we get very nice differences. For example, this is the reference variety Acteur in early June, and this was our composite cross in the same plot, just next to each other, and we could really see how the stripe rust that then died down because of the temperatures had really gone wild in some varieties. Of course, we have other references that stayed completely clean. The Belgians also say it stayed clean because their references obviously were resistant. And our modern materials, some of them have some very nice resistances. But I did get a call from Bioland in Germany asking me, can we do something? Can we spray? I said, too late. there is no thing that we can do in organic farming when we have rust. Either we have preventatively chosen the right varieties or use diversity, or we just have to live with it. Barley adaptation to extreme climates, multiple stressors. There are field trials in Finland and Denmark with spring barley were in 2013, and in Finland for diseases and for yield stability. And then there's greenhouse and phytotron trials with elevated CO2, nutrient deficiencies and pathogens in Denmark. And just to give you an idea of what it looks like, they have these open top chambers in the field where they put many, many different varieties. You can see those are variety plots. They have different environmental conditions. They blow in the CO2. Because CO2 is heavier than air, they can change the CO2 concentrations here, and they have different nutrient inputs. And so they look at, of course, phenotyping, marker associations, and studies of disease infections. And so you can see land races, cultivars, breeders' lines. They look for a phenotype in the phytotron in future environments. That's the idea. And then they go in. I remember what FAC is.
Yeah, CO2.
Free air.
CO2.
It's free. It's some. Yeah, it's open top chambers.
Oh, not exactly.
Yeah.
No,
not exactly.
The idea is the same.
Yeah, I think I wrote it down somewhere, but I forgot. This is just giving you an idea for how. So you have the different environments. You can fill me in for the different things, but there's definitely temperature, there's ozone, there's CO2 elevation. This should be the control, and then I'm not sure what it means, but what you can see, different genotypes react differently, and the Sebastian, they circled here because it basically did not really change according to the environment. And others really, you can see the CO2 went up like hell or nothing, or with the temperature it really decreased. Temperature is very important because it sterilizes in the wrong moment, and then you don't get fertilization, of course. Then we have evaluation of grain legumes for adaptation to extreme climates. That should be in Belgium, Luxembourg, Latvia. In Turkey, they're looking at linia and soybeans. There is, I just got some reporting that these trials are going on, but no results yet. And I'm also missing a couple updates still. But I know the stuff is ongoing, but we are talking the first year, so there is not much to be reported yet. Then weeds and intercropping. There has been a field trial of spring wheat in Sweden, Estonia. They looked at hydroponic and allelopathy studies on composite crosses. In Sweden, root hair studies, basically looking if the root morphology has something to do with allelopathic interactions and with wheat interactions. And they set up field trials of legumes in 2014. Based on the results from screening experiments in Belgium and Luxembourg. Some of them are also running at our research farm in collaborations with the Belgians, with the Luxembourgians, sorry. And that's what I put here. Those are the winter legume trials. They're running at our experimental farm in Domaine Frankenhausen. The Estonians have looked at baking quality in spring trials last year. So they compared composite crosses in selected lines. And when you go through here, the labels here, you see here is a composite cross. Here is also a composite cross. So this is the full loaf and this is the cut loaf. And what you see here is a mixture of different of these three, and here's the mixture of these three. And they are just checking what diversity will do, and I think it's rather convincing that the mixtures look quite good, and the composite cross also doesn't look bad at all, so they made some very nice testing on quality. Then they looked at early seed rigor at SLU, organic and conventional CCPs they received from us. We have kept the seed, we have maintained our composite cross populations under organic and conventional conditions, and have save the seed. We are now in the F14. Now F13 is growing currently. So they received seed of the F6 and the F12, so they have some 6 years difference, and looked at the early seed vigor. And there you see conventional versus organic. And the problem here is not so much a problem of the farming system as a problem of seed size and seed quality. And it has to do that we had a much worse soil in the conventional and than in the organic. And it reflects that we got not so good seed quality harvesting. And especially in the A and the Q, and especially in the F12, that was very strong because the F12 was the year where we had the severe overwinter kill. And in the conventional side, it was even worse than in the organic one. That is something they checked the 1,000 seed weight and said it's very clearly it's more a question of seed size. Although you could argue, of course, the seed size is also a question of the system, but normally we would have at least the same seed size, if not better, in the conventional system. So just looking at milestones and deliverables, the data from the spring sowing trials in year 1 should be finished. I'm quite sure from what I received that there's quite a bit available, and by the 18-month report we will have a lot to show. And then the trial, seed preparation, spring sown crops, year 2, of course, also has happened because the trials are in. And the rest has already been reported in the last executive board meeting. So all I can say, the work is amazing, and I'm really grateful to everybody that the response when we ask for reporting is so well done. Because with this many partners, this is actually a nightmare. And I was really not happy to become the coordinator of this work package, having another EU project to coordinate, but I have to say it's fun working, and so it's really nice. So thank you everyone.
Okay, thank you very much, Maria. That's great. Does anybody have any questions about the work package? I have one about the bread making. What bread making process were they using? Was that
I couldn't tell you. That's something I have to ask back. It looked like white bread to me. And we are also doing baking quality stuff. Sven, was this white bread or full? White bread also, yeah. So we have a little bit of that. So those are standard trials, basically. So we are doing it with the standard laboratory, and so it's white bread.
Okay.
I mean, this was clearly white bread.
Yeah.
But we will report a little bit in the talk I give tomorrow also.
Okay. Now I'm just interested because a project that I'm writing the report of at the moment, we've done some baking trials with the CCPs as well.
With white bread or with
white bread, yeah.
And how did that perform?
Very well in terms of for artisan baking. So hand baking, it got very good reviews, as good or better than the standard varieties. So
and in the laboratory baking?
Basically, we didn't have enough high enough protein content and the Hagberg falling number. It was not appropriate, essentially. Yeah. So artisan baking it works very well, but otherwise it just doesn't work.
I mean, most artisan bakers tell you that they can bake almost everything because you can adjust. Yeah. And the question always that we also pose is white bread baking the correct trial that we are doing? Because a lot of the organic materials are at least with some whole grain, more whole grain that is being used. And so there are also whole grain trials, but they're harder to come by. And so what we do currently, we do that with the white bread, but we are discussing also with Professor Klöger at our site if we should start thinking about some more whole wheat trials. But then what to compare to? We have done that before.
One question about...
how do you then standardise that?
Exactly. Yeah. So you have to know what mill has been used, what dough, and the main problem with the standard trials in the laboratory is that, first of all, the gluten contents, the amount of gluten you get depends on the amount of lactic acid you use to wash it out. And so you have to have a guess on the gluten that you have, and then you standardize against that. So if you have a high gluten sample, you should use more lactic acid to wash it out, and then a low gluten sample. So if you throw high and low gluten samples together in the machine that is said, okay, use that much lactic acid to wash it out, then the low ones get underestimated, well, or get overestimated because there's too much going through, and the high ones get underestimated, so the values get more together than they really are. At the same time, we really need to look at the different types of gluten, and that's normally not done in the standard trials. You should look at the high molecular weight and the low molecular weight glutens, and you should look at their relationship, because that has something to do with the baking quality later, much more than the total gluten.
What about using other tests like Chopin or Chopin?
Like what?
Chopin.
Farinograph or alveolograph.
So farinograph. Yeah, that would also be extremely interesting. But again, that's yet another set of tests that get very expensive, and so we always say baking is more interesting than everything else, because baking combines the dough properties with the gluten properties and all this. And all these separate values, we have basically said, okay, let's get just simply the protein contents and then baking. Because everything else in between is interesting but very expensive.
So we had another question in the middle.
Yeah, did you, with the industrial testing, did you actually do the baking of this composite cost calculation?
No, we didn't. No, we were basically what we did in that project was taking the different measures of the different variables that they needed. So looking at protein contents and Hagberg falling number and those things, and so it was never actually baked through an industrial process. So basically, it didn't meet the criteria for the bakers that they wouldn't take it anyway. So so it was but it never actually got to baking. So you don't know.
Yeah.
In that sense.
It is some baking trials.
But yeah, but not industrial. No.
No.
Yeah. Coffee is outside. We'll break now for 20 minutes.
Yeah.
20
start
minutes.
at past.
So if we start again at past, and just help yourself to coffee outside.